We need new ways to help people safely lose weight and to keep it from coming back.
Hello, I'm Sophie Roffner from the American Chemical Society. Welcome to this media briefing from ACS Spring 2023. Imagine getting the benefits of gastric bypass surgery without going under the knife. That's the objective for a new class of compounds that, in lab animals, reduce weight dramatically and lower blood glucose. The injectable compounds also avoid the side effects of nausea and vomiting, common with current weight loss and diabetes drugs. Now, scientists report that the new treatment not only reduces eating, but also boosts calorie burn. To tell us more, we're joined today by Robert Doyle from Syracuse University and SUNY Upstate Medical University. Thank you for joining us.
My pleasure.
Where did you get the idea to do this project?
To me, it was the obvious crossover of my two major interests, which are medicinal chemistry. I really like to make things in the drug sphere, but also endocrinology, which is really the body's way of controlling everything from sleep to appetite. And so the entire field was in need of a new route, a new approach to treat obesity, which has become a major problem in the United States and really worldwide. And so for me, it was just an exciting new avenue to explore, which is, why we do science.
How did you investigate this?
So that's a great question. So that not easily is the answer. The problem is we have to understand where the control mechanisms are in a human for appetite control, but also for how we expend our energy. And we have to do that without obviously disrupting the heart or making the patient feel ill. The last thing we want to do is essentially leave the patient sort of lethargic and tired, fatigued, feeling extremely nauseous. So we have to uncouple all of that while at the same time come up with a drug that can push the right buttons. And on top of that, we're very limited in the models that we can use to explore whether we're successful or not. Much of this cannot be done in a standard lab model like a rat or a mouse because they cannot vomit. So we can't measure human side effects accurately. And so we had to do this in the shrew, in a sort of a pseudo primate model that will allow us to tell whether we were even right or not. And so complex from A to Z.
And what did you find out?
So we found out that we could uncouple food intake from nausea and vomiting. That's actually really, really positive because they're all controlled by the same part of your brain. The sort of lizard part of your brain that controls appetite also controls gastrointestinal stuff like nausea and vomiting. And so we didn't believe we could uncouple those two, but we were actually able to show we could. And as a consequence, we produced a drug that not only produces really profound weight loss, and we were reducing food intake in rodents by 87% over 2 days, which is really phenomenal. And we were doing this at doses 10 times that of current FDA medications without any observed nausea or vomiting. When we translated it from the rodent food intake into the shrew emesis or vomiting study. So obviously really exciting and really, really, really positive for future development.
What mechanism might cause the reduction in eating? And do you think the weight loss is caused solely by eating less?
Well, food intake reduction is a big role. I mean, simply reducing one's appetite means less calories ingested, and which of course will reduce weight. However, that's not going to be sufficient if we want to target a population that need to lose significant amounts of weight and, more importantly, keep it off. And so step one, sure, is look at mechanisms that reduce food intake, but really we want to see changes in energy expenditure. We want to see increased locomotion, greater activity. We want to do all this without affecting the heart. And on top of that, we would love to be able to change metabolic behavior across the human as a whole, from the liver to the pancreas, so that you get greater metabolic comparisons to what we might call a typical lean person, right? Or the effects that you would get post-bariatric surgery, where we see changes in protein levels that seem to be the real reason why people keep weight off post-bariatric surgery. It's the sort of rebooting of the computer. And so we want to be able to do that reboot as well as just quote unquote simply reduce food intake, right? So a lot of wants on our list.
Have you seen any side effects, and how do your compounds avoid them?
So side effects, no. We have not seen any nausea, any evidence of nausea, again, in a rodent model. So by pica or the consumption of non-nutritious substances, or by conditioned taste avoidance, which is a Pavlovian response. We don't see that in either case. And in the shrews, we don't see emesis vomiting. So side effects, the ones that you would typically envisage, you would see in an FDA-approved current medication, we don't see any of those. So that's really nice. We also don't see locomotion changes or malaise or any evidence of lethargy. So again, in all testing we've done to this point, compared to FDA standard drugs, we do not see those side effects at all. So that's obviously hugely positive. The reason is complicated, and we're still figuring it out. But what we believe is we are actually targeting multiple receptors in the brain and in the pancreas and liver simultaneously. And it's the sum contributions of those receptors communicating to each other that is facilitating these changes. Really, what we want to do now is get down to the level of transcriptomics, so get down to the level of what proteins are changing, what neurons are firing. And to do that, we have a plan to do what's called 10x transcriptomics, where we can look at individual single neurons and look to see what genes are changing in response to our drug that are not changing in response to, say, a current therapy. And is that the reason why we're seeing what we're seeing in an empirical sense? So I think as a medicinal chemist, I want to go from A to drug and then work back to why it works sometimes. I mean, ultimately, I don't want to spend time working out why something doesn't work if I don't have to. It works. Now we figure out why it works. And then that will help us make it better or guide its development in the long term.
Do you have any data about whether the weight loss can be sustained after your treatment? In other words, would it be necessary to continue the treatment for the long term, or could it be taken for a while and then stopped?
So we have evidence in ongoing longitudinal studies, by which I mean out to 28 days, that we can sustain, and by which I mean continue to see a decrease in that food intake reduction and energy expenditure over time, which posits really well for the future in terms of being able to take this for a long period of time, lose a significant and profound amount of weight, and then with lifestyle changes and habit changes, sustain that weight loss, especially if we do rewire and endocrinologically reboot the human. I think that's really going to be the proof of the pudding in that aspect. But we do know that we can see compensation once we stop the drug. So we do know that if you stop taking this, it will wane in effect, but that we still see a maintained weight loss even when we stop it, which is not the case with current FDA therapies, where you will compensate pretty quickly if you stop taking the drug, unless you induce lifestyle changes, which is what most of these FDA-approved drugs will encourage you to do because of that.
What other applications might this have aside from what you've already mentioned?
Yeah, one of the interesting things that we noticed along the way was that the same receptors we were targeting, and these are little peptides that can target multiple receptors, but in concert, we noticed they were hitting the same family of receptors that have shown some benefit in reducing addiction behavior. So we actually pivoted and in parallel actually began looking at opioid addiction and fentanyl craving, and in particular fentanyl relapse in rodent models of fentanyl addiction. And what we noticed was we were actually able, with the same drug, to reduce the time, extend the time between cravings. So in other words, when the animal would go seek fentanyl, that time was greatly increased before the animal would go seek fentanyl. So in other words, this would help a patient who is trying to come off fentanyl, for example, or an opioid, and reduce that craving that is obviously driving the addictive behavior, the relapsing behavior. So obviously having something that can offset the actual at target craving effect, but then also delay the seeking behavior would be wonderful and something we can't do. And it turns out these drugs are doing that in fentanyl addicted animals. So that was a very unexpected and pleasant surprise.
Quite exciting. All right. Thank you, Rob, for sharing your research with us.
Anytime. Thank you.
Be sure to check out our other media briefings for ACS Spring 2023, which will be posted throughout the meeting at www.acs.org/ACS Spring 2023 briefings.